Hi everyone, welcome again to the course of structural biology. We are discussing structural biology techniques, high resolution, and this is the last class of this module of NMR spectroscopy, and today we will discuss about 2D and 3D NMR in detail and how those techniques are helping in solving protein structure. So, I was talking about routine 3D NMR experiments, the homonuclear through bond, heteronuclear through bond and through space correlation methods. Example of 2D homonuclear experiments through bond, COSY, the correlation spectroscopy, which also called homonuclear correlated spectroscopy. Its scalar coupling, as already talked about, it is through bond, identifies all coupled spin systems, correlate between protons that are coupled to each other. So, one H with one H. Then come TOCSY, total correlation spectroscopy. It uses spin lock for coherence transfer. During the spin lock, all protons of the coupled system become strongly coupled, leading to cross peaks between all resonances of a coupled system, and that is why it gives better results. Inadequate, the incredible natural abundance doubly quantum transfer experiments. Here it is 2D 13C 13C. Inadequate is useful for determining which signal arise from neighboring carbons, as we have already talked about. The 13 carbon signal is weak. However, it is very intensive as 0.01% of the carbon are excited at natural abundance. Use this experiment as a last resort when all the other correlations fails. So, little bit details about correlation spectroscopy COSY. Here both the axes correspond to the proton NMR spectra 1H and 1H. The COSY spectra indicates which hydrogen atom are coupling to another. The information on the hydrogen that are coupling with each other is obtained by looking at the peaks inside the grid. So, as we talked about already, there will be two axes, both 1H and 1H, and then we will look at the cross peak. We find a cross peak, we know that this hydrogen is interacted with that hydrogen. So, that is a simple process. These peaks are usually shown in a contour type format like height intervals on a map, but we will show it in a grid. So, this is a COSY plot. In order to see where this information comes from, we consider the example of ethyl 2 butanoate. So, if you see, you will see that there is a cross plot which is A, there is a peak. This peak indicates a coupling interaction between hydrogen at 6.9 ppm, which you will see here from the values that it's come here from 6.9 and another from 1.8. This corresponds to the coupling of the CH3 group, which is at 1.8, and the adjacent hydrogen which is at 6.9 in the alkene. So, this peaks talks about that. Similarly, the peak marked B indicates the coupling interaction between the hydrogen at 4.15 if you see, and the hydrogen at 1.25, which is here. This corresponds to the coupling of the methyl group and the CH2 group. So, you see here another peak. Very interestingly, if you see the diagonal in the symmetry, you will get another B peak as well as another A peak. Notice that there are a second set of equivalent peaks also marked A and B on the other side of the diagonal. Example of other 2D homonuclear experiments which are through space. So, now we are talking about bond, but these are through space. One noisy nuclear Overhauser effect spectroscopy. This is dipolar coupling, identifies neighboring spin system within 5 angstrom distance. It identifies chemical exchanges because it goes through space. This is single most powerful NMR technique for determining the 3D structure of molecules from conformation of small molecule to the 3-dimensional structure of small proteins. So, that is why noisy is very important. Roorkee rotating frame Overhauser effect spectroscopy. It is very similar to noisy, except that the initial state is different. Instead of observing cost relaxation from an initial state of Z magnetization, the equilibrium magnetization is rotated into the x axis and the spin locked by an external magnetic field, so that it cannot process because of the rotation here, rotation onto the x axis. It is called rotating frame Overhausser. So, little details about the nuclear Overhausser effect. NOE is caused by dipolar coupling between nuclei. So, there are nuclei like this one and this one. They are not connected through bond, but they are coming closer to space. This is very interesting because it will give you 3D idea. What I am talking about? Suppose you have a protein, this is a protein sequence, but when you want to understand the protein structure, you want to understand the fold. So, by getting this hydrogen and this hydrogen coming closer, you will get idea about which residues are closer. So, that is what NOE does. The local field at one nucleus is affected by the presence of the other one. The result is a mutual modulation of resonance frequencies. In NOE, the intensity of the interaction is a function of distance between the nuclei according to the following equation: I equal to A into 1 by R to the power 6, where I is the intensity, A is the scaling constant and R is the internuclear distance. So, if you see there are 3 hydrogens, the arrows denote cross relaxation pathways, R1 2 distance between proton 1 and 2, and their interaction are 2 3 distance between proton 2 and 3. The NOE provide a link between an approximate measurable quantity in the experiment I and the internuclear distance. As I told, by analyzing those distance, we could comment on the closeness of 2 groups which are not connected through bonds. NOE is only observed up to 5 angstrom, but this is for general. There are other systems where 3D NOE is performed, and we could get more than 5 angstrom. Examples of 2D heteronuclear experiments through bond and through space: HSQC, heteronuclear single quantum correlation or coherent spectroscopy. HSQC experiment is one of the most used experiments in biomolecular NMR, that is the determination of structure of DNA and protein, majorly protein. The HSQC experiment is one of the fundamental building blocks of scores of multidimensional heteronuclear and triple resonance NMR experiments. The HSQC experiment correlates chemical shifts of one nucleus to another. The 1H15N, the proton nitrogen pairs in amide groups of amino acids in proteins, are convenient reporters of each amino acids. The 1H15N HSQC spectrum of a protein is a fingerprint. So, when we are doing spectroscopy, we always look for fingerprints, because if we have some fingerprints, if we have some standards, it is very easy or very reasonable to do the analysis, because there are many spots in the spectra. So, if you know that this spectra belongs to that is very helpful. That is why HSQC is very important. So, in the 1H15N HSQC spectrum of protein, this is the fingerprint that can be used to monitor structural changes. So, you could study ligand binding, you could study variance of pH, temperature, a solvent, a salt, anything, and these are very critical experiments. For highest sensitivity, uniform 15N labeling is used, but for more concentrated samples, even natural abundant sample can be analyzed with modern high sensitivity instrumentation and cryogenic probes. So, you even do not need to label the natural nitrogen, is or sometime also be good enough with a high concentration sample. they are not at all limited to 1H15N, 1H13C important for organic chemistry as well as biomolecular NMR. One study was performed using HSQC experiment of a protein calmodulin in unbound state and bound state of a drug called W7. The HSQC experiment is, as I told, fundamental building blocks to record scores of multidimensional heteronuclear and triple resonance NMR experiments. Chemical shift of amide 1H correlated to directly bonded 15N for each amino acids. So, this is the drug W7, which is also called N6 aminohexyl 5 chloro 1 naphthalene sulfonamide. So, this is a typical sulfonamide. If you know, sulfonamides are typically used for inhibiting folate biosynthesis pathway. So, this enzyme is bound to calmodulin. The work is done by Ikura and co-workers, and if you see the black spots in the spectra is actually showing the unbound calmodulin, whereas when this drug W7 is bound, there is shift which is shown by the red spots. You will see clear spots, and this will help you to characterize the shifting of the amino acids. So, it is a very convenient way. You whenever you need, you have a drug library, you could screen it and you could take the help of HSQC spectrum. HMQC, heteronuclear multiple quantum correlation, information content compared to HSQC is more or less same. They are same type of process, only the single and multiple nuclei. The HSQC versus HMQC analysis of protein had made by Bax and co-workers to find out that the information kind of coming up are more or less same. There are differences based on the relaxation of multiple quantum magnetization as opposed to the single quantum. So, the information coming is same, but there are differences based on the relaxation. There are differences based on the dipolar broadening of multiple quantum coherence as opposed to the single quantum. There are differences based on unresolved coupling that broaden signals in the directly detected dimension. Multiple quantum magnetization does not evolve with scalar coupling, which could be used as an advantage in some cases. HMQC can be different from 1H15N versus 1H13C and the size of the molecule. HMQC can be subtle and difference depend on the application and the how the experimental setup is done. You could get similar but different effective information from HSQC and HMQC. HMBC, heteronuclear multiple bond correlation. The HMBC experiment gives correlation between carbon and proton that are separated by 2, 3 and sometime in conjugated systems 4 bonds. Direct 1 bond correlations are suppressed. This gives connectivity information much like a proton-proton COSY. The intensity of cross peaks depend on the coupling constant, which for 3 bond coupling follow the Karplus relationship. For dihedral angle near 90 degrees, the coupling is near 0. Thus, the absence of cross peak does not confirm that carbon proton pairs are many bonds apart. Also, there is Hector, which is heteronuclear correlation, usually correlate between 1H and 13C resonances mediated by the JCH coupling constant. So, proton NMR spectra on one axis and the 13C NMR spectra on the other. The Hector spectra matches the hydrogen to the appropriate carbon. The information on how the hydrogen and carbon are matched is obtained by looking at the peak inside the grid, as we have already observed for other ones. Again, these peaks are usually shown in a contour like format, like height intervals on a map. So, this is a Hector representation, and we are again looking at ethyl 2 butanoate. Here we are looking at again 2 peaks A and B. A is, if you look at, it is near the middle of the grid. This peak indicates that the hydrogen at 4.1, if you see hydrogen here and 13C here. So, it is around 4.1 and attached to the carbon at around 60 ppm. This correspond to the OCH2 group. Similarly, the mark peak B is indicate hydrogen at near to 1.85 is attached to the carbon at 17 pm. The grid value is not given here, but you could understand that these are from lower to higher. Since hydrogen, so if you see here from this is from lower to higher, the values are given here, the values are given here. So, you see from lower low to high. Since the hydrogen is a singlet, we know that this correspond to the CH3 group attached to the carbonyl in the acid part of the ester and not the CH3 group attached to the CH2 in the alcohol part of the ester. So, in that way it helps us to understand the relation. You could also notice that the carbonyl group from the ester has no match, since when the carbonyl part is interacting there is no hydrogen present. Another one, the last one is HOIC, which is Heteronuclear Overhausser Effect Spectroscopy. It makes correlation between proton and heteronuclei that are close in space like noisy and rosy. Now, we are shifting to 3D or multi-dimensional NMR. A 3-dimensional NMR experiment can be developed from a 2-dimensional one by incorporating an additional indirect evolution time. In addition to that, we also need a second mixing period between the first mixing period and the direct data acquisition. So, now you could include more indirect evolution times and you could add the dimensions. So, we talk about 1D NMR, we talk about 2D NMR and we are now talking about 3D NMR, but this is the same thing as you are understanding from here, how we are like taking 1D, making it 2D and then making 2D to 3D, then we continue in that way in higher dimensions. But 3D NMR like 1D, 2D, 3D are the most common practice. Each of the different indirect time periods T1 and T2 is incremented separately. There are two principal classes of 3D experiments. One, experiments consisting of two 2D experiment merging their results, the triple resonance experiment. So, one is if you have 1H and N15, you could get into another set of 2D experiment, but in triple resonance you need 1H, 13C, N15. So, that is why triple resonance experiments are really effective, but they are expensive. If you are considering protein, you have to grow your protein in presence of N15 and C13. So, they are expensive. There are many situations when the protein, the organism does not want to grow in presence of N15 and C13. So, there are lots of challenges. So, when we are taking 2D noisy HSQC or toxy HSQC, 2D spectra like noisy or toxy of larger proteins are often crowded with signals, because higher the size of the protein there would be more signal as well as less tumbling, you have to understand that. So, when we do NMR experiment, what happen, you put the molecule, be it a small molecule and a macromolecule in the solution, they tumble in the solution and depending on that the spectras are recorded. Okay. Now, if you consider a small molecule, it is always tumbling in a very high speed, so it is not a problem. For macromolecule like protein, when it increase its size, what happen? So, one protein molecule, a peptide molecule, it's tumble like a small molecule with higher molecular weight. What will happen? the tumbling speed would be decreased. This is very critical for NMR experiments of protein. So, when you have up to, I would say, 100 amino acid, 120 amino acids, they are okay, but with higher ones the tumbling become slow and that push towards lower resolution. One of the solution is you go for higher megahertz power. So, you start with 600, which is like minimum requirement for a protein NMR, go to 700, 800, 900, now you have gigahertz, 1.2 gigahertz. So, you increase, you get higher resolution, no doubt. Problem with high power, your temperature of the system enhance, and you already know from my like repeated demonstration that protein, what we are studying is about the folding of the protein, right. So, you cannot study the folding of the protein at high temperature, because at high temperature the protein denature. So, you need cryoprobe. Now, the cryoprobes, like more temperature they have to control, higher the price. So, overall you see the price of the experiment, the cost of the experiment enhance, that is one of the challenge. So, therefore, this spectra, so because they are crowded with signal in the larger protein, the spectra spreading out in a third dimension, usually 15N or 13C, so that the signals are distributed in a cube instead of a plane. So, you have two, if you 2 dimension, if you make it 3, so like 2 dimension, right. Instead you make this. So, basically what you make is this one with this one. Now, it is in the cube. So, you get a higher dimension, it is more higher resolution. This spread out is achieved by combining HSQC and noisy in a single 3D experiment. The noisy experiment is extended by an HSQC step. Acquisition start after this HSQC step instead of at the end of the noisy mixing time. The resulting experiment is called 3D noisy HSQC, which is very critical for protein. In a similar way, a toxy HSQC can be constructed by combining the toxy and the HSQC experiments. Now, coming to triple resonance experiment. The triple resonance experiments are the method of choice for the sequential assignment of larger protein greater than 150 amino acids. These experiments are called triple resonance because 3 different nuclei, proton, 13C and F like 15N are correlated. Now, here I want to tell you something. Somehow, if you have a other nuclei, just like in protein, when we are talking about, we are talking about only process where we directly go to 3D, we take the help of metal to phase them. Here, if the protein have a ligand or something, the ligands are having different atom, you could play in different dimension, and that is the beauty of NMR. That is where NMR is actually playing really smart. Remember the story of monkeys, that is we have to achieve there. People are working on developing these systems, some systems are already being developed. There are so many options of setting up different experiments, there are so many potentials that makes NMR a really great technique. The most important advantage of triple resonance spectra is their simplicity. They contain only a few signal of each frequency, often only one. The problem of spectral overlap is therefore markedly reduced. This is the main reason why proteins of more than 20 kilodalton can be assigned with triple resonance experiment. However, the coordinates of clearly separate signals from different amino acids can accidentally be identical, which is called degeneration of signals. We have to note that. The correct choice of connectivity between amino acids is the main problem in the assignment of triple resonance spectra. Another advantage of triple resonance spectra is their high sensitivity, which is caused by an efficient transfer of magnetization. The magnetization is transferred via 1J or 2J coupling directly via the covalent chemical bonds. The distance make the things weaker. Therefore, the transfer times are shorter and the losses due to relaxation are smaller than in homonuclear experiments. The disadvantage of all triple resonance experiments is the necessity of doubly labeled protein, the preparation of which is often expensive and challenging. There is a whole bunch of triple resonance experiments which cannot be covered in this one module of my study. So, I will explain only the general nomenclature of triple resonance experiments, and also I will talk about HNCA, which is the prototype for all this experiment. If you understand HNCA, you will understand others, because HN and C alpha, these are the important connectivity. But if you like to learn more, you could see the link here, link to obtain details about other techniques. Coming to the nomenclature of triple resonance experiment. The names of triple resonance experiments should little odd, when you see at first glance, but they are very descriptive. The names of all nuclei which are used for magnetization transfer during the experiment are listed in the order of their use. Bracketing the names of nuclei which are used only for transfer and whose frequencies are not detected. I will use two experiments as example to illustrate this, HNCO and HNCACO. So, as I told, HNC and HNC, but here C alpha is also used, whose effect is there to transfer, but its frequency is not recorded. HNCO experiment. In the HNCO experiment, the magnetization is transferred. You will see here, this is the I minus 1th residue of the protein and this is the ith residue of the protein. They vary with the R's. Okay. So, typical R's are recorded as we have talked about, but here you will see the transferring from HNI, which is here HNI, via the Ni, means ith residues in nitrogen atom, to the directly attached CO. So, this is the N, this is the H, and this is the carbon of the CO, and return the same way to the HNI, means this hydrogen nucleus which is directly detected. The frequencies of all the three nuclei are detected. So, if you see here, you will see these are the three nuclei, this is the hydrogen, this is the nitrogen and this is the carbon, all three its frequencies are recorded or detected. In the HNCACO, which is HNCACO, so this is again HNC, and here if you see the two carbons which is C alpha. Okay. This C alpha, when you are going from there to there, you go through this, but its frequency is not recorded. So, in the HNCACO experiment, the magnetization is transferred from the HNI proton via the Ni atom and the Ca nucleus, which is the C alpha I, to the CO of the ith residue carbon and back the same way. The C alpha atom, which are highlighted here in yellow, acts only as relay nucleus, its frequency is not at all detected. It is the only frequencies of HN, N and CO. So, these ones they are recorded. So, two carbonyls are recorded. This nomenclature has the advantage that the spectra can be easily imagined by their names. So, that is I was talking about. You look at the name HNCACO, you know that there is involvement of H, N and CO. Who are they? H, which is connected to the nitrogen, nitrogen which is connected to the one carbonyl and the CO is here used. So, the two carbonyls are measured, but the C alphas they are used for relay, but their frequency is not detected. Okay. So, in the HNCO an amide proton is correlated with the carbonyl atom of the preceding amino acid, whereas the in HNCSO the correlation to the intra-residual carbonyl atom is also visible. So, here you connect or get information between the two carbonyls. The HNCA experiment is the prototype of all triple resonance experiments. So, if you understand HNCA, you understand all possible resonance experiments. Starting at an amide proton, if you see here, you have the amide proton CONH, the hydrogen, the magnetization is transferred to the directly attached nitrogen. So, from this it would be transferred to this, which is measured as the first spectral dimension. Okay. So, you will measure hydrogen to nitrogen magnetization transfer. Then the magnetization is transferred to the C alpha nucleus, which is measured as second dimension. So, this is then going to the C alpha, which is measured as the second dimension. Afterwards, the magnetization is transferred back the same way to the amide proton, which is measured as the third dimension. In each step, magnetization is transferred via strong 1J couplings between the nuclei. So, coming to the continuation of the experiment, here we have the coupling which connects the nitrogen atom with the C alpha carbon of the preceding amino acid 2J. The coupling called 7 Hz is only marginally smaller than the coupling to the directly attached C alpha atom, which is 11 Hz. Thus, the nitrogen atom of a given amino acid is correlated with both the C alpha its own and the one of the preceding amino acid. Therefore, it is possible to assign the protein backbone exclusively with an HNCA spectrum. So, you are working here, but you could correlate with the neighboring C alpha. But usually more triple resonance experiments are needed because the cross signal of the preceding amino acid has to be identified and degenerate resonance frequencies have also been resolved. Coming to the assignment, we are talking about assignment, but just what is assignment? The aim of the analysis of NMR spectra is to extract all available information about interatomic distances and torsion angles. In the initial stage of investigation by NMR spectroscopy, each resonance must be associated with a specific nucleus in the investigated molecule, and this process is called assignment. The strategies employed for the assignment procedure depend on whether only a homonuclear 2D spectra are available which are unlabeled protein, whether 15 N heteronuclear spectra are available, 15 nitrogen labeled proteins, whether triple resonance spectra both 15 N and 13 C proton is already there, doubly labeled protein are available. But in general, the assignment can be divided in two parts: the sequential assignment of amino acids in the protein sequence, because you know the protein sequence, and the assignment of the amino acid side chain. So, first we develop the backbone and then we assign the, so that is the first step, and then we want to add the side chains R1, R2, R3 in that. Secondary structure determination by NMR techniques does not require a full 3-dimensional structural analysis, as you could see in the X-ray crystallography. So, that is also a beauty. Knowledge of the amide and alpha photon chemical shifts are in principle all that is necessary, but if this information is available, it is also considered, like when you see that the chemical shifts are available, it is we consider that nearly complete assignment of side chain protons are also available. While obtaining the sequential resonance assignments in a laborious task, The NMR method is perhaps the most powerful and certainly the most accurate method of secondary structure determination without a 3-dimensional structure. So, there are hierarchy in the architecture, but if you think when you are working on X-ray crystallography, I have talked about the process details. You directly have a protein, you crystallize the protein and your outcome is a 3D structure. Looking at the 3D structure, definitely you could determine the secondary structures, but the knowledge of secondary structure is not helping you to go to the 3D structure, you get the final result. Here when you do not have a 3D structure, you want to experimentally determine a 2D structure, NMR technique have very special acknowledgement. So, what informations are there and how we use that, we have already discussed about those things, but just we want to culminate the knowledge here how we are using them to solve the protein structure. The 3 bond coupling constant between the intra-residual alpha and amide protons is the most useful for secondary structure determination. It is directly related to the backbone dihedral angle phi, but unfortunately there is no 3 bond proton coupling that can be related to the angle psi. So, right hand alpha helix phi equal to minus 57 and 3JHAHN is 3.9 Hertz, right handed 310 helix phi equal to minus 60, 3JHAHN is 4.2 Hertz, anti-parallel beta sheet phi equal to minus 139, 3JHAHN is 8.9 Hertz, parallel beta sheet phi equal to minus 119, 3JHAHN is 9.7 Hertz, and left handed alpha helix phi equal to 57, 3JHAHN is 6.9. So, because we get those fingerprints as we were talking about before, now it is very convenient to determine the secondary structures. NOE, as we talked about, NOE is instrumental. A number of short less than 5 angstrom distances are fairly unique to secondary structural elements, and looking at them you could get an idea. For example, alpha helices are characterized by short distances between certain protons on sequentially neighboring residues between backbone amide protons, which is DNN. You could see here this is DNN, as well as between the beta protons of residue I and the amide protons of residue I plus 1, which is DBN. Helical conformation results in short distances between the alpha proton of residue I and the amide proton of residue I plus 3, and to a lesser extent I plus 4 and I plus 2. This I plus 2 and I plus 3, I plus 4 NOEs are collectively referred to as medium range NOEs, while NOEs connecting residues separated by more than 5 residues are referred to as long range NOE. Long range NOEs normally is weak, but it need like separate experimental setup or special condition. Extended conformation, for example, beta strand on the other hand are characterized by short sequential DAN means alpha n distances. You could see all the parameters here. The formation of sheets also result in short distances between protons on adjacent strands DAA, alpha alpha and DAN. So, for alpha helix, the sequential NOEs observed in 3D 15 and edited noise are indicative of alpha helix. You could see, as I was talking about, the distances, and when you measure them you see that there is a trend. So, 3.5, 3.4 d alpha n I to I plus 2 4.4, 3.8 d alpha n I to I plus 3 3.4, 3.3 d alpha n I plus 4 4.2, it is absent here, d n 2.8 2.6. So, you get the pattern, and also so you get the pattern of the value and you get the pattern which are similar for alpha helix and 310 helix. So, if you look at the secondary structure assignment of this protein, there is amide proton exchange rate with solvent water, which is filled with the diamonds here, these ones. coupling constant 3J HNA filled in alpha filled circles, you will see them here in filled circles, and sequential backbone DNN and DAN NOE connectivities are classified as strong, weak or absent and are represented by the thickness. You know, if you look at, you will see that how thick they are on the spot, and you could understand them of a bar connecting the residues in question. Medium range NOE connectivities D alpha n, I to I plus 3 and I to I plus 4 are drawn as line segment connecting the residue contributing to the observed cross peak if it present. So, you will see that the line residues, they are showing the connectivity. Coming to beta sheet, across strand NOE is observed in 3D 15 and edited noisy and 3D 13C edited noisy are indicative of beta sheet. Again, you see characteristics values when you have short less than 4.5 angstrom sequential and medium range proton distances in polypeptide secondary structures, and you get the pattern for D alpha n and the DNN. So, this is the structure of the beta sheet. It is the alignment of the 4 stranded beta sheet observed in GRX3 deduced from interstrand NOE connectivities. The amino acid backbones is represented in stick format with residue numbers indicated above the C alpha position. You will see the numbers. Interstrand NOEs are drawn as double headed arrows connecting protons, giving rise to the observed cross peaks. You see the double headed arrows. Slowly exchanging amide protons are identified in red. So, from there you could get an idea and assign and solve the or determine the secondary structure. For turns, sequential NOEs observed at 3D 15 nitrogen edited noisy are indicative of turn. Similar to alpha helix, shorter amino acid stretches connect beta strand. These are the similar value. You will see the value of turn 1 and turn 2. Now, if you see that in the beta turn there are type 1 have 2 types and type 2 have 2 types. So, these values are not separating them. To separate that, you have to go to the trends where you see that turn 1, turn 2, turn 1 prime and turn 2 prime and half turn all have their characteristic patterns. So, when you get the value in the table, you know that is turn type 1 turn or type 2 turn, but looking at the pattern you could ensure what type of turn it is actually. Amide proton exchange rates which we are talking about, this is also important. The regular hydrogen bonded secondary structure protect amide proton involved in them as evidenced by their significantly reduced amide proton exchange rates with the solvent. Although nearly all polypeptide amide protons are involved in hydrogen bonds in a globular protein, those in regular secondary structures appear to be longer lived. For example, after placing a lyophilized sample of BPTI bovine protease trypsin inhibitor into water, many amide protons are completely replaced with deuterium. So, here you use deuterium labeled water within 1 hour. Over the next several hours, the amide protons in the N terminal and then the C terminal helix also completely exchange. So, you make this experiment, you keep calculating, you get the spectra on time, you see that immediately many amide protons are completely replaced within 1 hour. Next several hours, amide proton on the N terminal and then C terminal helix also completely exchange, but some amide proton participating in the central anti-parallel sheet are still present after some months. So, this is very interesting and very additional experiment information where it is telling you which hydrogens are holding strongly. So, you keep making the experiments and you will see the replacement happened or not. If the replacement happened, they are relatively weaker. If they are still there, they are stronger in that way. Chemical shifts, as we have already discussed about that in detail, have very important role. Since the chemical shift of a nucleus is sensitive to the environment, it should also contain structural information. Correlation between chemical shift tendencies and secondary structures have been identified. The alpha proton of all 20 naturally occurring amino acids has been shown to have a strong correlation with the secondary structure. Wishart et al. Have produced a simple method for secondary structure determination by analyzing the difference between the alpha proton's chemical shift for each residue and that reported for the same residue type in a random coil. So, they are taking alpha proton chemical shift and then from the secondary structure, and they are comparing them with a random coil. Helical segment have grouping of alpha protons whose chemical shift are consistently less than the random coil values, whereas beta strand had values consistently greater. So, you could get the standard and you could differentiate them. In this way, the location of helix and strand segments are possible and quite reliable, although the boundaries of the secondary structural elements are difficult to determine. So, if you see chemical shift difference between C alpha C beta random coil values and experimentally observed values yield secondary structure chemical shift. So, if you see on the phi psi plot, you will see that the helix delta C alpha is 3 ppm, delta C beta is minus 1 ppm, whereas in beta strand delta C alpha is minus 2 ppm, delta C beta is 3 ppm, and this is the chemical shift of the entire protein. Selection of secondary structural segments. Unfortunately, no single criteria, coupling constant, showed amide proton exchange or short and medium range NOEs is sufficient for an unambiguous assignment of secondary structure. For example, short distance greater than 3.6 angstrom or greater than or less than 6 angstrom here between sequentially neighboring amide protons DNN are a necessary consequence of a helical conformation, but the presence of a short sequential DNN is not a sufficient criteria for helical prediction, as 49% of such occurrences are not in helical conformation according to Urtri's work in 1986. However, sequential stretches of residues with consistent secondary structure characteristics, NOEs, coupling constant, slowly exchanging amide proton and chemical shifts together provide a reliable indication of the location of this structural segment. So, it is not the one, but the combination of all the methods which will enable us to do that. Going from calculation of tertiary structure, the idea of computer aided structure calculation is to convert distance and torsion angle data, the constant, into a visible structure. However, the experimentally determined distances and torsion angles by themselves are not sufficient to fully characterize a protein structure, as they are based on a limited number of proton-proton distances. Only the knowledge of empirical input data such as bond lengths of all covalently attached atoms and bond angles enables a reasonably exact structure determination. To do that, a randomly folded starting structure is calculated from the empirical data and the known amino acid sequence. The computer program then tries to fold the starting structure in such a way that the experimentally determined interproton distances are satisfied by the calculated structure. So, you start with a protein sequence and then you start satisfying the distance criteria which you get from the experiment. In order to achieve this, each known parameter is assigned an energy potential which will give minimal energy if the calculated distance or angle coincides with its input value. So, this is what I explained very elaborately in X-ray crystallography, that is called refinement. In X-ray crystallography, because you are getting the structure directly, you have the software where you have the geometry and other informations together. So, you do energy minimization to go and set all the manually developed model. Here in the similar way, you develop a cutoff and you start assigning those parameters and you keep refining them. By refining I mean go to the minimal energy. The computer program tries to calculate a structure with a possibly small overall energy. Without the experimentally determined distance and torsion angle constants from the NMR spectra, the protein molecule can adopt a huge number of conformations due around its chemical bonds in the NC alpha bond and the C CO bond. So, this we have talked about. So, if you see in the protein they are connected with these. Now, though this is partially double bond. So, this is locked, but this is free and this is free. So, they are rotating and this is rotating. So, they could theoretically achieve millions of conformation, but they are restricted with the other factors which are coming from the constants, and that is why it is telling that without the experimentally determined distance and torsion angle constant. So, if you have those constants, then you could have fixed the model. All these possible conformations are summed up in the so called conformational space to get a optimized conformation. Therefore, it is important to identify as many constants as possible from the NMR spectra to restrict the conformational space as much as possible, thus getting close to the true structure of the protein. When it is X-ray, you just get informations, you connect the relations and you get the optimized structure. You might have multiple conformers in some of the atoms, but overall it is one of thermodynamically stable conformation. Not in NMR, because it is in the solution, they are not in fixed conformation, they are moving. So, you get a lot of conformations and you have to put the distance and restriction parameters, so that you could optimize to some minimum number of models, and that is why a big difference between X-ray crystallography structure and NMR structure is X-ray have only one model, whereas NMR have multiple model, you get a average of that. In fact, the number of constants employed is more important than the accuracy of proton-proton distances, so that the classification is sufficiently precise. So, if you see, I talked about the NOE class. For very strong it is 2.3 to 2.5, for strong it is 2.8 to 3.1, for medium it is 3.1 to 3.4, for weak it is 3.5 to 3.9, and for very weak 4.2 to 5. For calculation of tertiary structures, there are various computer programs employing two in principle different methods for calculating a protein structure. So, there are various programs, but they use generally two major principle. One is distance geometry or DG. This method is based on a calculation of matrices of distance constants for each pair of atoms from all available distance constants, bond and torsion angles, as well as van der Waals radii. This set of distances is seen then projected from the n dimensional distance space into the 3-dimensional space of a Cartesian coordinate system. So, when you take them, they go for infinite possibility. So, you have a n dimensional space and now you restrict that to the 3-dimensional space, that is the goal. This would enable the user to determine the coordinates of all atoms of the protein. Another one is simulated annealing. I have talked about simulated annealing before in X-ray. This is a molecular dynamics method which takes place directly in the Cartesian coordinate system. As I told, we have discussed this while talking about refining the crystal structures. In this method, a starting structure is heated to a high temperature in a simulation, that is the atoms of the starting structure get a high thermal mobility. So, what happened, if you remember, I talked about there are many possible maxima and many minima, right. So, when you are developing model, your model might be coming and restricting to any of these local minimas and your goal is to go to the global minima. So, you stuck here. Now, to get rid of that, what you will do? You will provide heat. So, it would go to above a threshold level and then you allow cooling, so that it would come and stabilize. How you then track it? So, what we do mathematically, we measure dy dx. Okay. When dy dx equal to 0, then it is either, so it is in a peak, either maxima or minima. Okay. Now, you perform d2y dx2 and depending on it is greater than or less than 0, you will determine it is in a minima or not. When it comes to a minima, you calculate the energy. Now, when you calculate the energy, suppose you get a arbitrary number, I am taking 10 kilocalorie. You do it next time, you get minus 8 kilocalorie. So, you know that this is a local minima because you have a better minima there. Now, the whole process, if you iterate, so perfect development of a computer program in a loop, you keep working and check the energy, you will probably find the global minima. During many discrete cooling steps, the starting structure can evolve towards the energetically favorable final structure under the influence of a force field derived from the constraints. So, how we will do the protein sample preparation? We will do the structure determination by NMR. Typically requires a protein concentration of 0.5 to 3 millimolar or greater, stable for several days at the desired temperature, usually 20 to 40, the experimental temperature. Proteins and peptides should be dissolved in a suitable buffer 10 to 50 millimolar with 10% D2O. Ideally, we would aim for an acidic buffer pH 4 to 7, but dissolve the sample in a buffer that provide maximum stability and solubility of the sample. As the sample needs to be stable for days, the buffer content plays a critical role in protein peptide sample stability. So, in NMR, which is not very much needed in crystallography, you should do a buffer optimization. Okay. So, now we have finally the NMR structures. The NMR structure of a protein is presented as a bundle of conformer. As you see, this is the representation of a NMR structure of SS10352 protein from Cynocystis species, and this is the crystal structure of the same. So, you see that the dynamics of the loop is higher. So, you get a lot of conformation. Each conformer present a good solution to the NMR resistant. First conformer usually is the base structure, and finally, typically a bundle of 20 conformers are deposited in the PDB. So, we started from a 0 concept in the periodic table, finding out a NMR active nuclei and then come to depositing the PDB. That was the journey. So, why we study protein NMR, why it is important? Initial days people used to think that we are studying NMR for proteins that are hard to crystallize, which was definitely true, which is true today also, but what is changed? We have found many other application of NMR structure for protein that can be dissolved at high concentration, to study dynamics of protein conformational equilibrium, folding and intermolecular interactions, for proteins which are drug target to screen a protein drug interaction, for DNA drug interaction. But honestly, with advent of new techniques, with advent of more sensitivity, with advent of more automation, possibilities are increasing. If you look at so many different type of interactions are already, you see so many different type of constructions of experiments, so many different setup and how differently they are giving information. This is definitely in a point from where we could go in such a state where we are getting more and more information regarding a protein. We will get information about more about the functional state of a protein, more about when a protein is binding to a substrate, more about transition state, more about its intermediates and what not. With that, I will finish this module, but before that I will go through a brief idea what we have learned through NMR. I will talk about brief ideas of NMR. We have the first idea that some nuclei have non-zero spin quantum number, making them NMR active. If current passed through a coil, it induces a magnetic field, and a changing magnetic field in the coil induces a current, vice versa. Placing nuclei with a spin I of half, where most of our like nuclei which are we are interested are there, proton, carbon and all, into a magnetic field leads to a net magnetization aligned along the magnetic field axis. When the B1 field is turned on, the external magnetic field, the net magnetization rotates down into the xy plane. When the B1 field is turned off, the net magnetization relaxes back to the z axis with the time constant T1. Individual spin precesses about the magnetic field axis. After magnetization is rotated into the xy plane by the B1 field produced from a pulse through the cell, it will precess in the xy plane. The individual magnetization vector whirling around in the xy plane represent a changing magnetic field and will induce a current in the sample coil, which has its axis along the x axis. NMR signal is a Fourier transform of the oscillating current induced in the sample coil. Nuclear spins produce small magnetic fields. Electrons are spin I equal to half particles, they produce small magnetic fields which oppose the external magnetic field. So, we talked about the nucleons are producing magnetic field because of the charge of the proton and the spin of the proton and neutron. Here electrons are also developing magnetic field which are opposed to the external magnetic field. The surrounding electrons shield the nuclear spins from the larger external B0 field. This results in a reduction in the energy spacing of the two energy levels and a lower Larmor frequency, which is called chemical shift. So, first one was the field generated by the nucleon. Now, here it is a shielding or deshielding effect. This is by electron, which is called chemical shift. In a frame of reference that rotates at the Larmor precision frequency, magnetization that is placed along the x axis does not move, it simply relaxes back to the z axis via T1 process. The nuclei with different chemical shifts and Larmor frequencies will rotate around the z axis at different speeds. T2 is the time constant for the magnetization vector to dephase in the xy plane. Idea 15, all the concept discussed above leads to different 1D NMR experiments. Combining them, higher dimension like 2D, 3D experiments are possible, which could be categorized into following groups: homonuclear through bond correlation, heteronuclear through bond correlation, homonuclear through space correlation and heteronuclear through space correlation. Idea 16, A higher dimensional NMR experiment can be developed from a two-dimensional one by incorporating additional indirect evolution times empowered with addition of NMR active nuclei if it is required. Like a 3D NMR experiment would be developed by performing experiment consists of two 2D experiment or the triple resonance experiment. Idea 17. The aim of the analysis of NMR spectra is to extract all available information about interatomic distances and torsion angles. For doing that, each resonance must be associated with a specific nucleus in the investigated molecule, which is called assignment. Idea 18. Analysis of 2D data would be performed by using chemical shift, nuclear Overhauser effect spectroscopy, coupling constants and amide proton exchange rates. Further, in dimension, computer aided structure calculation is to convert distance and torsion angle data constants into a visible structure, mainly working in the principle of distance geometry and simulated annealing. Idea 19. After solving the structure quality measures are performed using knowledge based methods like comparing existing crystal structure, MVD, measurement, RPF score, etc. using bond length and bond angle distribution, dihedral angle distribution, atomic packing, hydrogen bond geometries and other geometric features. Idea 20, the last idea. Now you started from scratch, you come to structure development, you have already refined your structure, you check the structure, finally the structure would be deposited to protein data bank using 20 conformers, first one with the highest resolution. So, this is our journey about NMR. As I told in the beginning of the course, this is not a course to teach you about the details of NMR. This is a course which will give you basic knowledge how NMR works, and I hope this will make you excited to know more about NMR, because as I have tried to talk through the course, through the module, that this is one of the next generation technique with millions of potentials. With that, I will finish today. Thank you very much.